World experts in gene editing have been visiting Cambridge to explain the significance and relevance to genome research of the technology CRISPR. CRISPR edits, removes or replaces any gene in the genome and has already been successfully used to treat genetic diseases. The intensive course, run by CAM Bioscience, stresses the importance of good practice to make the most of the technology. Holly Goodall reports.
Once the work of science fiction, now a reality. The genetic editing technique CRISPR is already transforming medicine across the world. By removing and replacing any gene in any genome, researchers and drug developers are working on treatments which could spell the end of genetic diseases.
Well, CRISPR is a bacterial immune system that we have adapted and applied to human and mammalian cells to make specific changes in the genome and the genetics. So with it we can apply it for all different kinds of approaches, such as gene knockouts, gene knock-ins, many different things that are now, with long thought to be implausible, are now essentially plausible. I think initially where we are now is it is allowing us to understand those diseases a lot more because we can build representative model systems. We will definitely see CRISPR also being applied in the clinic at some point in the near future too. First of all, there has to be some safety concerns addressed where the clinic's concerned, but I'm absolutely confident it will happen in the near future.
But with this powerful technology comes a responsibility for good practice. And it's this which the Cambridge startup company CAM Bioscience are hoping to inspire in participants on their super intensive genome editing course. With the technology developing at a rapid pace, the course brings in expert speakers to introduce scientists and researchers to the latest developments.
So we wanted to basically get as much information as possible about state-of-the-art, cutting-edge CRISPR technology, conveying best practice as far as we can about understanding how to do the edits properly, how to check that you've edited what you think you've edited, how to screen for potential off-target effects, so effects that happen elsewhere in the genome that are not as a result of your intended modification. Because the way CRISPR works is kind of like a search function in Microsoft Word. So you do your Control F, and that would essentially be your guide RNA and Cas9 protein. And then you can use that to find a particular gene or a word, if it's in a word processing program. And then once you are at that location, then you can do your edits. You can add a word, you can delete a word. So it's very specific. It finds only the sequence that you're interested in and nothing else. And that's the idea. But potentially, if you don't design your guides properly, maybe you're picking up things elsewhere. If you don't design the genes that you want to put in correctly, then maybe you actually end up cutting again and destroying your potentially new bit of genomic information. So there's lots of skills and techniques that scientists have to pick up.
The event's also a rare opportunity for industry leaders to meet with scientists and researchers to share ideas. Among the international companies represented, CAM Bioscience's official partners and equipment and tutelage providers, Eppendorf and Merck.
It's actually been a very different way of working for us. Obviously, we're a kind of industrial, we're an industry. This is academia, and the two have met, and it's happened in a kind of very informal way. So we've learnt quite a lot as we've gone along, and it's been a lot of fun, but at the same time we've been able to, it's incredibly important, I think, for the networking side of things as well, for both parties.
I think this is a great initiative to connect the industry and companies like ourselves with young research academics. You know, it's great for not only collaboration, giving us ideas about where we need to be, to fill those gaps in the experimental and lab workflows, knowledge transfer, and also gives us the ability to come and showcase our products and pipeline.
CRISPR technology still has its limitations and is undergoing development all the time. But it's hoped that in the near future this technique will allow scientists to overcome diseases previously thought to be incurable. Holly Goodall, Cambridge TV News.